What this research has meant for plant breeding is that we are able to be more efficient, our rate of genetic gain is improved, so it means that the varieties that we produce are better than they otherwise would be if we didn't have this research and the software we have to manage these many alleles.
If you have a look at our now collaborator AGT makes around 1,000 crosses a year. So if we could get predictions accurate enough so that they could then tell for all those individual crosses which was the most likely to succeed, that would greatly improve their plant breeding programs. So that was the major objective, was to get predictions that were accurate enough to do that and put them in software that was easy to use so the breeder could run many, many crosses and then decide which ones were most likely to succeed. Well, the stumbling block at first was to assemble big enough data sets so that we could make estimates of the effects of all the genes simultaneously instead of 2 by 2. So to do that we needed to put together, to amass really large data sets. That was what we thought. And so it was finding the data sets, getting the collaboration of people and putting them together from different places. The next step was to take the data out of the databases in formats that were acceptable to the statistical analysis programs, and I used GenStat. And so we had to develop query systems in the database which would handle the data, put it into the correct formats for statistical analysis, and then we ran it from there into the statistical analysis and then back out of the statistical analysis into the access databases to put in software which would make the predictions. So it was a multi-step process. I was aware that the multinational companies in the United States had prediction software like that. I wasn't aware of the details, I just knew that they had it. We discussed through the 90s trying to get it from them and decided that that was impossible. So we decided to do our own. Originally the software that made the predictions was developed at the University of Queensland by Dean Podlich. That software used the glutenin genes which have a very large effect on quality in wheat, especially dough quality characteristics required for making bread, noodles and biscuits. We had 6 genes at that time, and if my memory is correct, about 21 combinations of variants or alleles of those genes.
My contribution to the project was to bring some molecular biology capability directly into the team. Previously, the molecular analysis had used protein markers and was carried out externally. Now we have the capability to use diagnostic DNA markers within the project to fill the data gaps that we have directly.
Well, the biggest breakthrough was finding that the accuracy of the predictions was highly comparable to what had been done before in lots of experiments where the genes had been handled one by one and the alleles usually 2 by 2. But we'd done multiple alleles across multiple genes simultaneously, and ours very closely matched good experiments from the past. And we were very, very happy with that result when we first saw it.
For plant breeding, this means that breeders are able to devote less selection pressure to traits such as quality traits. They can expend their energies in other directions because we can now predict the results of their crosses using the data sets that we have. And we will in future be able to extend this to other traits, other disease-resistant traits or development traits, and we're only limited in what we can do by the number of diagnostic markers that we have for the different genes that would be involved.
The early work in wheat had been making predictions for grain quality. Now we tried grain yield, which is much more difficult because a lot more genes are involved. What we showed in barley was, yes, you can make, using plant breeding data, accurate predictions of the effects of genes on yield in barley. We now are trying to extend that to wheat, using the genes that affect vernalisation and photoperiod. As with quality, they have multiple alleles. We're working currently with 4 genes, and we hope we can predict heading date and ultimately grain yield using the same procedures we use for quality. That is very large plant breeding data sets and accurate diagnostic typing of the genes. There's no reason why it can't be extended further and further for any crop and for any characteristics of economic importance, whether it's quality, disease resistance or grain yield. The critical things are to have large plant breeding data sets and diagnostic markers for the genes which will accurately identify all the alleles or variants of those genes.